Thank you. And hello, everyone. Welcome to another week of the Town Hall. We have some opening remarks, and then we'll go into the questions received prior to today's call, then open the line. We are still seeing a significant number of applications every week. And then as a monthly total for COVID EUAs, pre-EUAs, amendments, and supplements, we're seeing still around 160 to 200 applications a month. And this is keeping us very busy, especially since the surge in staff for COVID has largely returned to prior to surge capacity and workload per reviewer. So the surge folks have gone back to regular Medusa work, which does include COVID pre-submissions, Q submissions, and full submissions for COVID assays. That does leave us back to a lower level of staffing for EUA. And I just wanted sponsors and developers to know that. We're doing our best to keep up with the volume, and largely we're clearing applications weekly at approximately the same rate that we're receiving them. So we should be cycling through on a pretty routine basis. So, but because we have fewer staff, our times for responses and everything may go up a little bit. This is unfortunate. We know we're doing the best we can. We ask for your patience. On the other hand, we've now authorized over 380 unique submissions and well over 500 supplements and amendments to date. So there is quite a significant number of tests available to the market. And we have seen testing go down across all categories in the U.S. And also the number of positive samples is way down. That's number of positive samples and percent positive rate going way down is great news for the country. I just want to put all that in context. All right, and our priorities remain the same as most recently previously stated. Diagnostic tests for point of care and home collection, and high-throughput central lab tests. I will turn it over to Toby. Thank you.
Thanks, Tim. Thanks, everyone, for joining us today. I have one update. On Friday of last week, we issued a safety communication regarding a Class 1 recall for the Lipu Medical Technologies SARS-CoV-2 antigen and molecular antibody tests. These were tests that had not been authorized, cleared, or approved by the FDA, and the company is recalling them under a Class 1 recall because they were distributed as unauthorized tests. We are aware that they were distributed to pharmacies to be sold for at-home testing by consumers, as well as offered for sale directly to consumers. So that safety communication and Class 1 recall were posted on Friday of last week. And then I can move into the handful of questions that we received ahead of time. The first one we have is related to comments that were made on a previous town hall recommending that for antigen and molecular tests, data subset should be provided in the EUA submission for vaccinated individuals enrolled in the clinical study. And the questions being asked are for a point of care and over-the-counter EUA requests for a lateral flow antigen test. What data should be collected from the enrolled vaccinated subjects to be provided in the data subset? And if a vaccinated subject is confirmed positive or negative with both the candidate device and the EUA authorized highly sensitive RT-PCR comparator method in the clinical study, can the vaccinated subject be considered part of the confirmed positive or negative samples needed for the EUA submission? To address those, first we would ask that the data be included in the clinical line data as additional columns and should include the date of vaccination, which vaccine was administered, and if it's a multi-dose vaccine, the number of doses and the dates that they were administered. And then it may be acceptable to include all subjects together, provided that they're not pooled retrospective and prospective specimens. But we do want to see the performance stratified by non-vaccinated versus vaccinated individuals. And depending on the specific situation, we may request additional presentations of the data, such as 1 dose versus 2 doses. And we would also want to see asymptomatic and symptomatic stratified as well. Our next question is about pooling of saliva specimens for a previously authorized EUA assay. This company specifically has a PCR assay that has been authorized for use in either respiratory or saliva specimens, and they are looking to seek authorization to pool up to 5 saliva specimens. So they're they're asking about the template, which states that a high sensitivity real-time PCR should be used to confirm that at least 25% of the saliva specimens pooled are low positives. And also asking about whether they can use archived PCR positive saliva samples. So we did recently authorize the first saliva test for pooling, and that is the Clarify COVID-19 test kit by Quadrant BioSciences. And the publicly available decision summary for that EUA outlines the study details for a saliva pooling approach. So we would recommend you take a look at that to get a better sense for what we are looking for. And in that document, you will note that saliva samples were tested individually and in a pool to establish performance, and no further comparator method testing is needed to establish a pooling claim for a previously authorized EUA test for saliva. We also note that archived samples are acceptable to establish a pooling claim. And then the last question that we have is from a company that is preparing a pre-submission for a multiplex PCR gastrointestinal assay, and asking, based on the previous feedback about priorities, whether that would fall into one of the categories identified as priorities by FDA. So again, those priorities are COVID-19 related, companion diagnostics, breakthrough designation requests, and a significant public health impact. And those relate to the pre-submissions that we will be reviewing. So as we've discussed previously on the town hall, those are the categories or types of IVD pre-submissions that we intend to review at this time. Tests outside of those categories, we are not likely to have the resources to review right now. So we will be generally declining other IVD pre-submission requests. And at this, based on the information provided in this question, it does not appear that this multiplex PCR gastrointestinal pre-submission would fall into one of those categories. There are, however, other GI multiplex 510K cleared assays that can be referenced for the recommended analytical and clinical study designs. And with that, unless Tim has anything additional to add on any of those questions, we can open the line up for the live Q&A.
I say we open it up, take the live questions.
Great. Thank
Thank you. We will now begin the question and answer session. If you would like to ask a question over the phone lines, please press star one from your phone, unmute your line, and speak your name clearly when prompted. Your name is required to introduce your question. If you would like to withdraw your question, press star two. Again, to ask a question over the phone lines, please press star then one. One moment as we wait for any questions. Our first question comes from Annie Wright. Annie, your line is open.
Hello, thank you for taking my question. My question again is about the requirement for a mobile app after EUA approval for an OTC COVID-19 product. We just wanted to confirm because we're in the process of developing a OTC product, and we realized that there are requirements for mobile apps for reporting. So we're currently working with a mobile app contract manufacturer right now, supplier, and we just wanted to confirm that the main requirement that the FDA would like to see for the mobile app is basically the ability to report to health authorities only. Are there any other requirements? We'd just like to confirm that.
Yeah, so thanks for the question. Just want to clarify that this is not a requirement to make the initial authorization. We have authorized a number of OTC tests that didn't yet have a mobile app for reporting. The intent here is to assist in managing the public health crisis. And therefore, what we're asking is an AMP that can report positive and negative results to public health authorities, state, local, and national. So that's the intent, and what we've been doing is arranging a post-market commitment for those developers who don't have a reporting feature at the time of original authorization.
Okay, thank you so much.
You're welcome.
Our next question comes from Ron Domingo. Ron, your line is open.
Hello, thank you for taking my question. FDA has previously focused on testing using fresh samples for supporting antigen tests. However, with the growing complexity of obtaining fresh positive samples, the sponsor that we're working with would like to freeze samples collected during a post-EUA study, then use the frozen samples to support development of another assay for an EUA submission. We're seeking FDA's feedback on this, on an alternative approach to address the issues of obtaining sufficient positive samples at this stage of the pandemic.
We recommend that you check with us on your plans for banking for antigen tests and to use frozen swabs, maybe direct swabs. We're open to that pathway when enough positives can't be obtained in a prospective study quickly enough. We still want to see a prospective study, at least see the negatives and see the performance there, and confirm performance in sites prospectively. And hopefully you'll see, well, not necessarily hopefully, but maybe you'll see some positives in that prospective study. And then we're very open at this point to different methods of enriching those positives. And however, the details could be very specific to a given device, so we would ask you that you address these questions through a pre-EUA, or if you already have a pre-EUA, to add that to the list of questions that you're asking the reviewer. So a couple of things to consider is how do you source and select those positives, and is there introduction of any selection bias that could impact evaluating actual test performance. For example, if you were to use a very insensitive test to select those positives, you would be biasing your test population to higher positives. And we want to try to see the full spectrum of positivity from low amounts of virus to high amounts of virus, and, in your, depending on your device, but for the claim period of time for your device. And then also we have seen, particularly for antigen tests, where a freeze-thaw cycle does increase the sensitivity of the assay. And so we would want to see some sort of freeze-thaw comparison study to a fresh sample. And there's multiple ways to do this with banked samples or other methods. So do check with, do propose something and check with the FDA team. And we would hope that there would be no bias introduced by the freeze-thaw. That is, the test performance would be the same, whether it's frozen prior or not. And then, if there is a difference, assessing that difference and the impact on the study design. So those are some of my high-level thoughts. I don't know if Toby has anything else to add.
No, nothing to add.
All right. Thank you for your detailed response, Tim and Toby.
Our next question comes from Franco Calvarez. Franco, your line is open.
Thank you for taking my call again today. My first question is related to the notice that came on Friday about the LIPU tests. And there was a mention that these tests were distributed through some pharmacy networks. Is there any further detail as to what pharmacies or what distribution networks those tests were made available through? Because we're talking about 8.4 million antibody tests and at least 205,000 antigen tests, and so that's one. And the other one is a question related to the previous question. So we have been doing a lot of due diligence studies that would help us understand better the clinical performance of our test. And to do that, we purchased, I believe, about 30 positive samples with CT values. And these are nasal pharyngeal samples with various CT values. We have found very positive concordance in general. Now, is there, given that the prevalence is low, as it has been pointed out many times, is there any way to use those samples as part of the official clinical validation, or would we have to basically start from zero after submitting the protocol for the clinical validation to FDA? You may be able to use that data, at least in support of your submission, and I would suggest you come in with a pre-EUA to explain what you've done and ask that specific question. The things we would look at is how did you obtain those samples? Is there any bias? What was the comparator test used? Was it an acceptable comparator test? Is the range of positivity acceptable? And then was there any impact of the banking freeze-thaw on performance? And was your protocol set ahead of time before you did this evaluation? And there was another thought. And you haven't changed your device. And then the people who read the device are your intended use population. So there's a lot of questions there. But bottom line, is it a sample type that you're seeking authorization for? Did
the population that you're intending to be used for the test, perform the test, and interpret the test. So those are some of the questions, and there's a lot of questions there to address to find out if that study is acceptable for use in support of your application. So again, I would lay that all out, ask the team through a pre-EUA. If you've already submitted a pre-EUA, you can go back to the same reviewer and come in with these new questions. Toby, anything to add?
Not on that aspect. I can respond to the first question about the safety communication for the LIPU tests that went out last week. The information that is posted on our website is all of the information that we have publicly available right now for the distribution of those tests.
All right, thank you.
Mm-hmm.
Our next question comes from Howard Ernovitz. Howard, your line is open.
Hi there. Hello, everybody. I wanted to find out if, in fact, there's any guidance or guidelines that I can find that is regarding the next generation sequencing, laboratory developed test, if you will, for long COVID, that is the post-COVID individuals. We're going to plan a study. We have some preliminary data that we may have a tool that could be diagnostic. And I'm wondering, in planning the study, is the number still going to be 30 symptomatic long COVID patients, looking at 30 asymptomatic long COVID or post-COVID at this point, and then perhaps 30 healthies? And thank you.
So just some follow-up questions. You said laboratory developed test, so this would be a test designed and developed and validated in a single lab or within a single healthcare system?
Yes. Yes.
And the test you're going after is prediction of a long COVID sequela. Is that the purpose of the test?
Yeah, the purpose of the test is to make a diagnosis that the gene sets that we are, the first gene sets that we're looking at right now, can tell the difference between people with active COVID infection and people that have recovered and are asymptomatic. And so the genes involved in this make sense. They're all a very specific immunologic set of genes. So one single lab making a diagnosis that you, in fact, have a gene set that is more likely to be not recovered or COVID related, and then use it also as a monitoring test to see if, in fact, combinations of steroids or the like can be used in a liquid biopsy. So the first application would be diagnosis and confirmation.
Diagnosis of long.
Yes.
Okay. So I'll address the technical question. I may throw the LDT question over to Toby. So, and this is for anybody interested in this sort of test. Certainly, this has been a topic, but we haven't authorized such a test. And I'm not sure that we're ready to provide recommendations on the validation for those that are interested in developing such a test. So at that point, I think I want to throw it over to Toby. Toby, if you're ready to catch this one on LDTs, what our current stance on LDTs is.
Sure. I think there's not a whole lot that we can say at this point about LDTs. That is still an issue that we're working through. But we do still have the guidance document out that discusses lab tests as well as the HHS statement from last year.
Yeah. So the bottom line is the FDA has stated that they're not currently reviewing LDTs, and nothing's really changed on that. And I don't know that we can add anything else. And then developers of this kind of technology that do want to, say, kit it in a traditional manufacturer, I would just say that if you're thinking about this, that they come in through a pre-EUA and engage in a conversation with us. I'm sorry I couldn't be a little bit more encouraging of providing some information here at this time.
Good, thank you.
Our next question comes from Ojun Suk. Ojun, your line is open.
Thank you. Yeah, I just had a quick question that's similar to some of the questions that were asked before regarding the clinical evaluation of a molecular test for the EUA with the positivity rate and prevalence going down. We already have a study designed and plan to start for prospective collection, but our test specifically uses a direct swab as a sample type to be used for testing. So I was wondering if we wanted to go down the route of complementing the prospectively collected sample data with the bank samples or contrived samples, if it's going to be acceptable to use contrived samples that have not been banked or diluted in the VTM or other matters that are usually done for frozen samples and bank samples. And if we can just use those contrived samples near our limited detection to supplement or to complement our prospective data, or if we have to specifically find a banked sample with a known CT value that is somehow in a derived format. I don't know if that's possible, and use those for to support the prospective samples collected for our study.
So I think what you're saying is what happens, or maybe it's already happened, you've already done your study and you're not getting a lot of low positives. And what do you do if you don't get enough low positives? And can you use bank samples in some way? Is that correct?
I guess just to clarify, I'm just asking, given the low number of positives and positivity rate, we could be having trouble collecting all 30 prospectively collected positive samples. So again, related to the questions asked earlier during the Q&A session, we were also thinking of potentially supplementing the data, the prospective collection data, with the banked or frozen samples. But I was wondering if we can use the contrived samples as a set of supplementary data to support the prospective collected data if we cannot get all 30 positives within a reasonable time frame. And if we do use the contrived samples, if we still have to do a comparator testing on the contrived samples.
Yeah, I'm not sure what you mean by contrived, because you're talking about banked or frozen. So
I guess just to, again, clarify, there are two different types. So, I mean, one approach is to use the bank samples, but another approach I was thinking of doing this was just to use a contrived sample with the virus particle spiked into the negative swab. Again, given our test is a dry swab, I was thinking making a contrived sample at a very low LOD, or close to our LOD, would be more straightforward than finding the banked samples that would be, amenable to be used with our testing protocol. So, not sure if I'm making any sense here.
Well, I just wanted to be clear. We're not currently entertaining contrived samples. We, as I mentioned at the top of the hour, or at the top of the call, we've authorized now over 380 EUA authorized tests, and going back to contrived samples would be obviously something That's quite challenging, especially in the face of testing demand and testing utilization going down. So we're of the mind that there are more than 30 banked samples out there, that we're open to using bank samples if you've attempted a prospective collection and been unable to achieve the recommended number of positives. And then if you're going to move to banked, we recommend that you check with our review team to make sure that process of going to banked is done in the least biased way, and the proper validation is done around the use of banked and frozen samples.
Okay, thank you.
Our next question comes from Roseanne Chan. Roseanne, your line is open.
Hello, hi, thank you. Yes, I have a general question. We haven't heard about high sensitivity COVID RT-PCR test. Is this list of tests on the FDA website, or what is the definition of a high sensitivity COVID RT-PCR test?
So we do recommend that you do check in with the review team during your pre-EUA, or ask a very specific question into our email box for your test, whether or not a particular central lab molecular test is sufficiently high sensitivity. We're looking at those tests that are towards the high end of sensitivity and have a separate extraction/concentrating step before going into a PCR. And an RT-PCR assay is important because we're looking at the CT values and that comparator to assess what is the range of viral loads in your study set, so that we can evaluate performance across the dynamic range, not dynamic range, but among the expected level of virus in a usual population.
All right, thank you.
Mm-hmm.
Our next question comes from Ron Domingo. Ron, your line is open.
Hi, thanks for taking my question again. This is related to some previous comments and questions. We talked about the dropping positivity rates and rising vaccine rates, and sponsors think that it's going to be difficult to achieve the sample size required by FDA during the post-authorization studies in a timely manner. So what other options are available to the manufacturers for achieving post-EUA numbers? Would the agency consider reducing the sample size or restrict studies to a fixed period of time? Or would you also consider data collected outside of the U.S.? Thank you.
And so this is conversion of an EUA authorized assay to a full authorization assay?
Yes.
So I have been encouraging recently that you come in with a pre-submission. It's not a pre-EUA, it's but it's actual usual Q-sub pre-submission. We are accepting them for COVID, and we will address those questions. And, because of the current situation, we'll be as flexible as possible on that situation. And we are looking through all possibilities using actual patient samples from banked to samples collected internationally, and even the testing performed internationally, if that's the best way to validate the test. But again, come in through the pre-submission and ask those specific questions to make sure that then, depending on the characteristics and the design of your test and where you're intending that test to be used, so that we give the most specific feedback we can on your particular device. But now's the time to really convert assays, and it's something I probably should have mentioned beginning of the call as well, is come in with those pre-subs and Q-subs now, and be working on your conversions if you want to stay in the market past when the declaration might be ended at some point.
Okay, and Tim, if it wasn't a conversion submission, would your recommendation still be the same?
So you come in through, if it's not been EUA authorized yet, then come in through a pre-EUA and ask about alternate ways of getting a positive. And again, we want to see a prospective study as we would recommend, and it's only when you've demonstrated that the inability to collect all the positives that we would look for alternative methods to accumulate more positives in your study.
Okay, thank you.
Our next question comes from Ashwood Doman. Ashwood, your line is open.
Thank you very much. My question relates to decisions made by the FDA late last year, around October, November. I don't want to make this about any particular number of companies, but some companies received deprioritization notices, including those that had rapid antigen tests with potential for at-home applications. I was wondering if these notices would require these companies to reapply, or have their EUAs been automatically put back into the applications line? Thank you.
So in general, it depends on the reasons for the deprioritization. But if it was, home tests and point-of-care tests, they're still a high priority, so it would not have been deprioritized for that reason. So sponsors would have gotten some sort of information from the FDA on why we declined to review or declined to issue. Those are the two main deprioritization reasons. And if a developer is able to address those concerns and wants to come in with a subsequent submission, they're free to do so. The FDA's concerns, though, would need to be addressed. Otherwise, the result may be the same.
Okay, thank you.
And I think to clarify a little bit about that question, if the test, sort of otherwise appeared to meet the priorities, I think you mentioned a rapid antigen test with potential for at-home use, they may have been deprioritized not because they didn't meet priorities, but because there was something lacking about the submission. And so I think that's what Tim was really getting at with needing to look at the specific reasons that were given to the company so that the company can address those issues prior to resubmitting. So for example, if it has potential for at-home use but was not validated for at-home use, then that would be a potential reason why a test may have gotten one of those letters. And we would want to see that validation completed prior to resubmission.
Or there could be some kind of performance issue or
right.
Inadequacy of the studies or design or something. So again, that response from the FDA is specific and tailored to each developer, so that the developer can decide what to do, address those in an appropriate manner and come back in or not. But we endeavor to be very clear and transparent in our feedback to each of the sponsors.
As a reminder, to ask a question over the phone lines, please press star then 1. Our next question comes from Lenny Tao. Your line is now open.
Hello, cannot hear you.
The line is open.
Hello? Can you hear me?
Now we can hear you.
Oh, yeah, okay, thank you. Yeah, I think my question has been answered. Thank you so much.
You're welcome.
We have no additional questions in queue at this time.
Let's hold for 1 or 2 minutes, and if there are no questions, we can end the call early. And again, thank you for everybody calling in and for asking these questions. Hope that we have been transparent and been able to clarify and provide specific guidance or recommendations.
We have another question in queue. Our next question comes from Elizabeth Brunley. Elizabeth, your line is open.
Hello, I just wanted to clarify something that I thought I heard on the call earlier. Did I hear you correctly when I heard that you thought that maybe the EUA would be terminated next month?
No, I'm not sure where that came from. Toby, do you know?
Okay, I must have heard something wrong.
No, not at all. I don't think that we would. I don't think we said anything about that.
So,
okay, thank you.
We don't think that the current emergency declaration will end anytime soon, which means that the EUA pathway will remain open and EUA authorized tests will remain available. Should a time come when it comes time for conversion to full authorization, the FDA will provide guidance and a pathway, and allow, reasonable time to come in with a full submission. All the while allowing the EUA authorized tests to remain on the market so that we meet our nation's testing needs. As far as I know, we're nowhere near that, and the guidance, our draft guidance hasn't been made public yet, and as soon as we can, that'll be made public so that all developers understand what they're looking at in a post emergency declaration situation. But we're not in my estimation, we're not anywhere near that. It's going to be a long time off.
Okay, thank you for the clarification on that. One other question: are you still recommending that if an EUA modification and/or submission was made weeks ago, that we follow up with an email and include you and Toby on the email?
Occasionally we do give that advice. I'm sorry, what was the topic again?
No, we submitted a modification to an EUA that was submitted last year, and then an additional EUA to couple with that for home collection several weeks ago. What would be the best way to follow up for that?
If you've been assigned a reviewer or a contact, you should have been. That may be the most recent submission if it's within 2 weeks. But you should have a contact, and you can reach back out to that contact. If you're not getting
you do not have
any
a reviewer. Okay.
You have a contact. You have a name of an FDA person.
No. Well, I have somebody that we used last year, but not somebody that was assigned this year, no.
Yeah, so I'll go back through the templates email box and try to seek clarification. And if they cannot clarify for you the status, then ask for your email to be forwarded to Toby and me.
Okay. Thank you.
Our next question comes from Dana Hummel. Dana, your line is open.
Thank you for taking my question. We would like to get the FDA's feedback on our proposed clinical study protocol. However, we have not submitted a pre-EUA yet. Would it be okay to request feedback on our protocol before we submit a pre-EUA?
That would be the purpose of the pre-EUA, is to put the protocol in there and any other details about your assay for us to evaluate your protocols. Anything that's clear from our templates doesn't need to be asked, and it's only things that are outside of our recommendations, and if you want to confirm the comparator you're using is a suitable comparator.
Okay, we were just still compiling all the information for the pre-EUA, but we wanted to get started on the clinical study, so that's why I thought maybe we could just get your feedback on the clinical study protocol before we put together the entire pre-EUA.
It's probably more efficient for you to get everything together,
Okay.
and then our team has everything to review. You're going to want to have that feedback before you start, and so. Looking at just one part of a potential pre-EUA, isn't necessarily the most efficient way to go about this.
Okay, perfect. Thank you.
Our next question comes from Franco. Franco, your line is open.
Thank you for giving me the opportunity to ask a couple more questions since there appears to be a light audience today. So the first one is regarding a saliva control step that we have been doing at our own discretion, really, because we have been iterating the sensitivity of the antigen test. So since the Abbott BinaxNOW, the Quadrant QuickView, and a couple of others can actually be bought now directly from pharmacies, we actually got our hands on a couple of Abbott BinaxNOW, and I believe the CARE test from Access Bio, and we used them as positive control with the specimens that we purchased, which were in CDC VTM, and I wanted to also provide a little additional detail about how we obtain those. So basically, and this would address the issue of the bias, perhaps. So basically, we hired a lab that has been doing a lot of testing through the pandemic, and they use a high sensitivity PCR test with an extraction step. So we knew that from the get-go. So basically, what we did for those 30 samples was we said, hey, when you get a positive, let us know. We like to test that person to see, how our test performs, and that's in effect how we ended up getting those 30 specimens. So would that be a biased way? So that's what is related to my first comments regarding. Are you aware of the? Sorry, may I continue?
Yeah, but hold on a second though, because this is getting very specific advice for your specific submission, and it's not something that we typically do on the call.
I apologize.
So the best way though to get because in all likelihood I need more detail than you're giving me right now to address bias. And so the usual way for us to do this is actually see the study protocol. How are you acquiring samples in the context of your specific assay and use case? So those are all important to assess, the acceptability of what you propose. And it takes a little bit more time to assess than we can on this call.
Sure, I apologize. Thank you.
No reason to apologize.
Our next question comes from Sri Kosik. Your line is open.
Hey, Tim, Toby, can you hear me?
Yes.
Oh. Hey, thank you very much for taking my call. You guys are wonderful, and the DMD group needs to win some kind of award for the EUA nightmare that they're going through. But I have a question about pre-EUAs. We submitted a pre-EUA template about 6 months ago and got a response about 2 weeks ago. If you're planning a clinical study for a EUA, for a SARS-CoV-2 EUA, and if we submit a clinical study to get your opinion on using banked samples, what is the timeline for response? Do you have a clue of, is it like 30 days, 60 days, 90 days, or whenever it comes, it comes? It's the reason I'm asking it is because we want to. Sorry, go ahead.
No. I cut you off. Go ahead.
No, I was just going to say the reason I've asked is because we have to get the clinical sites up and running, and there's a lot of planning that needs to go into place, and it would be useful to sort of understand what kind of timelines we're seeing for pre-EUAs, when specifically questions regarding how to do the clinical study, or is archived samples okay?
Yeah. So the team is working on how best to use archive samples right now. So hopefully on a subsequent call, I can provide more global harmonized information. For now, yeah, and if this is a priority submission, if they don't get back to you with an idea within 2 to 3 weeks, ask the templates email box to copy Toby and me, and we'll work on getting you faster response. As I said at the beginning of the call, we remain very busy, and we're back down to basically non-surge staffing right now.
Yeah, and that's the reason why I think really DMD is doing a wonderful job considering the fact that you guys are dealing with so much. So I really appreciate the reviewers for their time and your time too, but even we are strapped on our side too, so I know you understand that. Thank you.
Yeah, we've received over 5,500 applications in a year, including COVID and non-COVID applications, with 300, well, actually after 300 staff. So it's a large volume.
Yeah, it's an amazing thing. Thank you.
We have no additional questions in queue at this time.
I think we can go ahead and close the call, operator. Thank you.
Thank you for your participation in today's conference. You may disconnect at this time.